each speaker to declare any potential conflicts of interest before delivering their remarks. So first up, we have Angelica Chawla.
And I will unmute myself. There we go. Hi, I'm Angelica Chawla. I am an employee and a shareholder at Bluebird Bio, and I lead the clinical development program at Bluebird Bio for lovo-cel. I'm also a practicing pediatric hematologist oncologist for more than 20 years. I have dedicated my career to the care of individuals and families impacted by sickle cell disease, and today I'm more hopeful about their future than I ever have been before. Sickle cell disease is a complex and progressive disease that impacts every aspect of life. People living with sickle cell disease often face frequent vaso-occlusive crisis, mounting and irreversible organ damage over time, profoundly diminished quality of life, and ultimately a life cut short by decades, not years, but decades. And as bad as that sounds, from my experience supporting and learning from patients, the reality is far worse. The pain and fatigue that people with sickle cell disease endure and manage on a daily basis has impacts beyond the physical into mental health and psychosocial well-being. I have worked with children who are bullied and ostracized because they were small, weak, and had frequent absences from school. I've worked with young adults who, after multiple, unpredictable, and long hospitalizations, isolate themselves and be isolated by others as they were a burden to their families, friends, and coworkers. I have met parents who have refused opioids despite excruciating pain due to concern they would jeopardize their ability to care for their children, and others who live in constant fear they will not survive to see their children grow up. Even with all the advances of the last few decades, people living with sickle cell disease need and deserve options that they can have a transformative effect on their health and quality of life. Options that enable them to determine their destiny, rather than having it defined by their disease. As the most deeply studied gene therapy in the development for sickle cell disease, we are confident in the value that lovo-cel can deliver. First and foremost to patients and their families, but also more broadly to the healthcare system and society. In our studies, we observed that after treatment, the production and persistent presence of the modified anti-sickling hemoglobin A in the red blood cells of all patients to the last patient visit as far out as 8 years. Also shown is an improvement in total hemoglobin and complete resolution of vaso-occlusive events in most patients. These results signal the potential to improve the disease trajectory without the need for ongoing disease-modifying therapies. We see that these physical and clinical improvements are translating into improvements in quality of life, particularly the intensity of pain experienced by the individual and the overall impact of pain on the fundamental aspects of daily life. Further, we're seeing improvements in the ability to work and on work productivity. As ICER's assessment recognizes, all of these data points are consistent with a potentially transformative benefit for patients. As we discuss the potential value of lovo-cel for sickle cell disease, I cannot help but think of patients who have died waiting for these therapy options. I think of the time, effort, and sacrifice of these sickle cell warriors, their families and friends, not to mention the money they spent on medical care that couldn't keep them alive or free from vaso-occlusive pain. I think of Duran, age 20, an engaging college student with his own YouTube channel who voted for Obama proudly from the intensive care unit and succumbed to a stroke a few weeks later. I think of Paula, age 24, an effervescent and kind woman despite having severe chronic pain and recurrent vaso-occlusive pain, a hip replacement, and kidney failure. She had over 20 admissions annually for the last few years of her life before dying of severe vaso-occlusive event. I think of Ray, age 26 and a father who had a matched sibling donor but was unable to proceed with transplant and died suddenly from acute chest syndrome. We need more options for treatment with people for sickle cell disease, and for those with severe disease, we need them now. The decision of whether or not to pursue an option, a one-time option like this, should be in the hands of the patient, their family, and their healthcare provider, armed with information of the known risks and benefits, as well as an understanding of what is yet unknown. We are grateful to the sickle community who has been with us on this journey for close to a decade. We're both humbled and proud to be part of this new wave of potential one-time treatment options that this community both needs and deserves. In closing, I want to thank ICER for their thoughtful consideration and recognizing the value of lovo-cel. I also want to thank you for your time and the opportunity to share my perspective.
Thank you. We are going to have time after all of the comments for questions at that point.
Rena, we can take them now.
Okay. So open to any comments or questions for Dr. Chawla. I'm not seeing anyone on my screen, so unless I'm missing you, I think last chance we will move ahead. So our next speaker is Jamie Rubin Cahill. Excuse me. Jamie.
Thank you. I'm Jamie Rubin Cahill. I'm an employee of Vertex Pharmaceuticals, and I lead the Health Economics and Outcomes Research Department there. Thank you again for having me here to speak to the incredible potential of exa-cel to be a one-time transformative therapy for sickle cell disease. I'm going to focus my comments today in three main areas. One, the burden of this disease on people with sickle cell, their families, and society as a whole. Two, the data supporting exa-cel as a potentially one-time transformative therapy. And three, why the data we've reported on exa-cel are unique and cannot be directly compared to data on lovo-cel. Sickle cell disease is a life-shortening disease marked by painful blood vessel blockages known as vaso-occlusive crises, or VOCs, that cause severe pain, organ damage, and a range of other debilitating acute and chronic complications. These profound impacts are exacerbated by systemic healthcare inequities faced by people with sickle cell, including longer emergency room wait times, undertreatment of pain, and stigmas associated with the disease. The economic burden of sickle cell on the healthcare system is also profound, with recent estimates of lifetime costs for people experiencing recurrent VOCs totaling upwards of $4.2 to $6.2 million. The economic model produced by ICER underestimates these costs by using the lowest available VOC event cost in the model. In order to accurately value transformative therapies for this disease, healthcare costs must be appropriately considered. Currently available treatment options require chronic therapy that do not alleviate key aspects of sickle cell disease burden, especially painful VOCs. We now have the ability to change that outlook. Last month, we presented data on 35 patients with sickle cell disease who received exa-cel, who were followed from 2 to 39 months. 17 of these patients were evaluable for the primary and key secondary endpoint at the time of the data cut. 16 of 17 achieved the primary endpoint of freedom from severe VOCs for at least 12 months. The one patient who did not meet this primary endpoint did not experience any hospitalizations related to severe VOCs, a key secondary endpoint. Moreover, all of the additional 18 patients dosed with exa-cel were VOC-free in the evaluable period with mean total follow-up of 8.55 months. These results are in stark contrast to the approximately 5 VOCs that patients experience per year based on natural history data. Patients also reported substantial clinically relevant improvements in health-related quality of life across a range of domains, and the safety profile was generally consistent with myoablative conditioning and autologous stem cell transplant. Together, these data demonstrate clear differentiation of exa-cel versus current standard of care. The clinical and safety profile of exa-cel is further bolstered by data in a different disease area, transfusion-dependent beta thalassemia, where 48 patients have been treated with exa-cel using the same gene editing and manufacturing process and have been followed for up to 43.7 months, with almost 90% of evaluable patients achieving transfusion independence. These data on exa-cel are unique and cannot be directly compared to data on lovo-cel, primarily because of differences in how events were captured in their clinical trials. The exa-cel trial assessed severe vaso-occlusive crises, or VOCs, whereas the lovo-cel trial assessed severe vaso-occlusive events, or VOEs. Even though the two endpoints sound similar, in reality, they are very different. Severe VOC is more rigorous and inclusive of the events experienced by patients and results in a higher bar to achieve the primary endpoint. For example, severe VOCs include any pain event that requires a visit to a medical facility or administration of pain medications or transfusions. In contrast, a severe VOE includes only the events that required a hospital or emergency department visit that were at least 24 hours or at least 2 visits to a day unit or emergency department during a 72-hour period. As a reminder, 100% of patients treated with exa-cel met our key secondary endpoint of hospitalization-free for 12 consecutive months, which is a closer analog to the severe VOE endpoint. In addition to the differences in the clinical trial endpoints, exa-cel has a unique mechanism of action. Exa-cel uses CRISPR-Cas9 technology, a non-viral and extremely precise tool to edit relevant stem cells. Biologically, there is no known mechanism whereby these permanently gene-edited cells could revert back to their prior form, and therefore lifelong durability of treatment is expected. Durability we've demonstrated in the data reported so far. Other gene therapies, including lovo-cel, rely on viral vectors that randomly insert into the genome to edit cells. 20 years of data on lentiviruses have shown there can be a risk of malignancies caused by these random insertions. In contrast, extensive preclinical characterization of exa-cel has shown zero evidence of off-target editing, and with exa-cel, there is no virus and no random insertional mutagenesis. We have dosed over 80 patients who have up to 43.7 months of follow-up in sickle cell disease and TDT and have observed no malignancies.
Thank you, Jamie. I think we're going to have to stop right there. Any comments? David?
Yes. Thanks. I just wanted to respond to the concern about whether we were undervaluing the cost of VOCs prevented. And the issue there is, first, that there's a wide range of data on what those costs are, but also those costs are typically healthcare costs if people are hospitalized or go to the emergency department or are seen by a primary care doctor or don't seek care and stay home. And when Praveen was discussing the numbers he used, and we increased the number of VOCs from 4 to 5.1 per year, that was to recognize that some of these aren't treated and to make sure that we captured the disutility to patients who were having VOCs, even if they didn't go to an emergency room or hospital. That means, though, that we're averaging those costs across those $5,000 to $6,000 of cost per VOC for some VOCs that would not cost anything to the healthcare system. They'd cost things to patients, but not the healthcare system. And so balancing that out, we thought that given where we increased the number of events to, that the cost per event was reasonable. Certainly, this is an arguable point, but we think we picked the best number that we could given the data we have available. Thanks.
Thanks, David. Any other comments? Okay. Our next commenter does not appear to be here. That's Eve. And so we are going to move ahead to Maggie Delaski.
Hi, my name is Maggie Delaski. I'm pleased to be here today. Nothing additional to disclose from what's on the slide. I'm the Director of Advocacy at Sickle. Sickle is a national advocacy organization for sickle cell disease. I would like to thank ICER for this opportunity to be here today and provide comment on the recent evidence review. Sickle engaged closely with ICER during the 2019 review for sickle cell and provided a great deal of recommendations and strategies for how ICER can improve its patient engagement methods. We are pleased to see that many of these suggestions have been incorporated into ICER's new patient engagement program. Throughout the engagement with ICER during this review, we've noted several key limitations to ICER's abilities to reflect patient-centric dimensions within the cost-effectiveness modeling. Today, I would like to reflect on 3 limitations from our perspective. The first concern is that the model does not bring a truly comprehensive view of what matters most to individuals living with disease. As noted by ISPOR and many other economic experts, capturing patient-centric dimensions of value is crucial to properly demonstrate the value of new interventions can have. For sickle cell, many patient-important outcomes are omitted from ICER's cost-effectiveness model, despite strong and repeated emphasis on their importance from patients, clinicians, and manufacturers. For several years now, Sickle has been working to identify what matters most to those living with the disease through quantitative and qualitative methods. In a recent project led by Sickle, we worked with a multi-stakeholder group to prioritize value elements for sickle cell disease. From this project, the top 2 value elements were ranked as mental health effects, such as depression and anxiety, and overall quality of life. Neither of these are included in complications in the model, indicating the inability to reflect the full patient experience and priorities in the value calculation. In response to public comments, ICER notes that their discussion with experts led to the selection of conditions used in the model. However, there's a lack of transparency into who was consulted and if patients and caregivers were included as experts to weigh in on these modeling decisions. Sickle served as a liaison during ICER's focus group sessions with 4 patients and 4 caregivers. However, those sessions primarily focused on the patient perspective paragraph of the report and did not include members of ICER's economic modeling team. The second limitation highlighted by the community in Sickle was the use of inappropriate health state utility score data to reflect patient experience. Health-related quality of life weights that are used to generate the qualias incorrectly assume uncomplicated sickle cell to be 0.8 utility value. The reference study, Annie 2012, did not measure uncomplicated sickle cell. Instead, this study captured quality of life through the EQ-5D 1 week after patients were discharged from the hospital after having a severe pain event. In the article, Annie notes that patients were not completely pain-free at discharge, or importantly, at that 1 week follow-up. Health utility scores derived from this study are not appropriate or sufficient evidence to support a conclusion as a baseline for how individuals experience quality of life without pain. The third concern is about the perpetuating existing disparities using ICER's current value assessment framework for this review. As stated by Dr. Powell-Hayes and Dr. McGann in their 2020 article, there may be no population of patients whose healthcare and outcomes are more affected by racism than those with sickle cell disease. Within the background section and the patient perspective sections of the report, ICER cites realities such as poor healthcare quality, poor clinical communications, barriers to access, and poor uptake, which show how racism manifests across healthcare for sickle cell. Without the use of an equity-informative economic model, ICER is unable to adequately account for these disparities in their base case results, leading to concerns that recommendations from this analysis will contribute to further inequities. While today's committee deliberations will discuss equity considerations, the impact will be limited. Evidence from experts such as AMCP show that payers do not prioritize factors like equity in decision-making unless value elements can be quantified within the cost-effectiveness analysis. Today's committee will also be asked to vote on if there's adequate evidence to demonstrate health benefit. Meaningful data that clearly defines and measures effectiveness in this framework requires years of investment in time, money, partnership, and academic expertise, which has not been available to the sickle cell community for generations. In ICER's approach to be objective and consistent across all assessments, cost-effectiveness findings are unable to address the severe unmet need that exists for some conditions, such as sickle cell. ICER is retrofitting a framework that is not suitable for sickle cell disease, which perpetuates the mistreatment these patients know all too well. Thank you for this opportunity to comment. We look forward to continued partnership with ICER and other stakeholders working to improve lives with sickle cell.
Thank you, Maggie. David.
I just have a question for Maggie. Given your concerns about ICER's method on this, do you think that the price we're suggesting for gene therapy is too high or too low?
My comments were not focused on the pricing. It was focused on the economic model and the way that the model was structured to center those patient-centric dimensions.
Right, but the whole point of the model, of course, is to try to figure out a fair price for a curative therapy, potential curative therapy for sickle cell disease. So I guess I'm just wondering whether Sickle and you feel that ICER is suggesting too high a price for these therapies or is suggesting too low a price for these therapies?
I'm not making a suggestion on whether it's too high or too low. I'm trying to indicate that this model and the way that it's framed has limitations and should not be used to execute decisions in the current state.
Okay, thank you.
Okay, thank you very much. Oh, I'm sorry, Steve.
Oh, thanks. I just wanted to thank Maggie and Sickle for participating in the process and for helping us, as she said, learn from the first go around. And as we continue to go through that, I just wanted to also say that some of the aspects, I mean, you raised several important points, or maybe I'm going to forget some of them, and we're going to come back to some of these issues in the policy roundtable. For instance, I'm not sure right now that the United States has a quantitative weight that reflects how much we as a society would pay extra to address historical disparities due to racism. There is clearly an interest in understanding that and in thinking about it and talking about it. As far as a number to put on that right now, you could probably get very different focus groups in different parts of the country. You could get different political perspectives on this. And so in some ways, what we are hoping to do is to highlight that issue and to make it clear that this is a societal question that deserves more discussion. But we did not feel like it was our role to be the one to put a number on that. And I don't think you have a number to put on it either. But we really agreed, I think, very much that this is an issue that our country has to continue to view as certainly distinctive in many ways to sickle cell, but also that permeates across other kinds of conditions that might also be among patient groups that have suffered from historical disparities for a variety of reasons. So I'm glad you raised that issue. I just wanted to say that, again, we don't feel like there is one number that reflects the United States view on how much extra money to spend in one way that might address a historical disparity, but we're going to talk about it at the policy roundtable.
Thank you. I'm going to move us to our next commenter, Jeannie Cottrell.
Hi.
Hi. Go ahead, Jeannie.
Okay. Hi everyone. My name is Jeannie. Basically being, uneducated with sickle cell. On sickle cell and feeling something I never had to classify. The thoughts were how I could make sure Taliba didn't have to feel excluded because of her sickle cell. With the hospital visits and the pain, I never wanted her to feel labeled as just a child with sickle cell. Her spleen was removed at the age of 13, and accepting the fact that she had an illness because she made it 21 more years than they said. There were times when she wasn't treated right, disregarded, disrespected, and watching her go through it all, I sometimes felt I was looking for more than what was offered. And I was determined to do what I was allowed to do. Our relationship was strong and close. She knew I had her back. She knew I would, she knew I had her back, and she knew I wouldn't let anyone mistreat her or take advantage of her. There were times when the hospital staff would not respect her and do the things that they should do for patients. She would be judged as stereotype, and people would assume she was in the hospital only for medicine, when overall she really didn't even like it. I don't think I ever took the time to think about what sickle cell meant to me. I just wanted Taliba to live the normal life that she deserved to live. As her family, we all provided what she needed with the support of her going to the hospital multiple times with excruciating pains. Sickle cell never really occurred to me as being something that I had to address as I felt like I hated it, or I felt like I was robbed because it took Taliba home. Basically, when Taliba passed, the day before she did, she came to me. I was at her house and she was looking at me and she said, "Mom, you're always here." And I said to her, "I'm going to always be here, Taliba. Who else is supposed to be here? It's me, you, and the family. We have your back 100%. The next day, my daughter went home. So that's why I said, I don't know if I feel robbed or that I can say that I hate sickle cell, because we never ever look or live like she had something that should stop her from hindering her life of living. She was very talented, voice opinionating. Everyone adored her, a giver, a helper. And I just wanted everyone to know that she is greatly missed. And they told us that she will only live to 21, and instead she lived to 42. So I'm grateful to God that I had the opportunity to have her for 42 years. She will always be my sickle cell warrior. She will always live in my heart. She will always be with us. We always were going to be honored for the doctors that took great care of her, Dr. Gay, Dr. Kim Smith, Dr. Frimpon. We love them all. I just wanted everyone to know that you can't hate something that is a part of someone that you love. You have to live the way you should live, like nothing, like there's nothing in the place of it.
Thank you so much, Jeannie. I
Thank you.
speak for myself and I'm saying you have me very teared up. It's
I'm
really,
sorry.
really moving. And thank you for sharing all that.
You're welcome. This was a little challenging, but I had wrote a whole speech, but I couldn't do it. I just wanted to talk from my heart because I just know that she is in a better place. She is. She's not dealing with no needles, no doctors, no medicine, none of the things that she didn't like. So I'm grateful to God for that and for the 42 years.
We're very grateful to you for being here. Really, really it takes a lot, and I just want to make sure I allow anyone else to make any comments, but you've really moved us.
Thank you. Thank you.
Okay. Don't go away, Jeannie, but we're going to move to our next commenter, Tesha Samuels.
Good afternoon, everyone. My name is Tesha Samuels. I am a patient advocate. I am a wife, a mother, a friend, a daughter, and yes, always a sickle cell warrior. I was diagnosed with sickle cell disease, SS, at the age of 2. And Ms. Cottrell used a word that I had never thought about before, but she used the word robbed. And I would say that having sickle cell disease, it robs not only the patient or the warrior, but it robs the community and the world abroad of the talent, of the personality, of the perseverance, of the testimonies. My journey is not dissimilar to some of the other warriors that you have heard from today, but each warrior's journey is different. I was robbed of being able to attend school when I was in elementary school, high school. I was robbed of that communication and that socialism that you get with being a traditional high school student. Why? Because sickle cell had me in the hospital for months on end. It seems like from November to April, I was in the hospital at least for 2-3 weeks at a time each month. So it robbed me of that opportunity to get the education that I sought. Additionally, when I moved into adulthood, it robbed me of an opportunity to complete my college education. It robbed me of many days of being at the job where I wanted to be because I wanted to strive to be the best professional. We are taught that we are to be contributing members of society. However, we feel that we are left behind in society because of the way that we are treated, because of the way that the stigma that comes along with being a sickle cell warrior. I mentioned the many titles that I claim. However, what I didn't mention was some of the titles that you are given by society and the doctors and the physicians and some of the caregivers within the community. I was labeled a drug seeker. I have had to contend with many, many drugs in the market to try and reduce the amount of pain. I have also gone through therapy where I was on hydroxyurea for many, many years. I also, because of the over 250 blood transfusions, developed a condition where I needed iron chelation medication. And that was for 8 hours of my day, once a month. To add to that, you're talking about 4 to 6 hours for blood transfusions once a month, just to get the strength to be able to do and be a productive member of society. I underwent the Bluebird Bio treatment in 2018. And from that time, I am not saying that it was not challenging, but the challenges that you all have mentioned in some of your testimony today is in no way compared to what I had to go through as a sickle cell warrior for 36 years. In that 5 years since that treatment, I am a productive member of society. I am on no medication. I have not experienced a vaso-occlusive crisis. I have been able to enroll in school. I am a professional, and I add and contribute to my community at large. So when I tell my story, I am a daughter, a wife, a mother, a friend. And this type of opportunity really gives me so much hope for what it will be like for sickle cell warriors in the future. And so I am here speaking with you, and I'm thankful for the opportunity to do so that you can hear my story, someone that has actually been through it, to say that my life is forever changed. And I'm hopeful that it will continue to be so and be able to provide that opportunity for others like myself. Thank you for your time today, and thank you for listening.
Tesha, am I saying it correctly? Tesha? Okay, that
Yes.
was really fantastic. Thank you so much. I wanted to see if anyone wanted to comment or had any questions for you. Was your daughter, or did you say daughter or son? You said you're a mother. Were your children born before the treatment?
I have children in love. I was unable to have children, but my husband has children and they are mine.
Steve, did you have a comment?
I could say it now, later, or at the end of the meeting, and I probably will all three, but I just want you to know, you and the other folks who are taking the time to make these public comments honor us. And I just want to thank you very much.
Kim, I don't see your camera, but I see your hand there. Oh, and unmute.
Can you just clarify when you said you went through the bio treatment, what you meant by that?
Yes, at NIH, I underwent the Bluebird Bio level cell treatment 5 years ago. Yes, ma'am.
And the experience leading up to that, you alluded to it, but could you just comment a little bit about what that was like?
Absolutely. Leading up to the Bluebird Bio, so I was receiving blood transfusions on a monthly basis, just to be able to keep up with the amount of effort that I needed in order to contribute, like I said. So I was working a full-time position at that time, and I really couldn't even get up a flight of steps. And so in order to really be productive, I needed not only the pain medicine, but I needed to definitely get my blood transfusions on a monthly basis. Also, prior to that, I was hospitalized for at least 2 weeks, probably 6 months out of the year. I hope I
Thank you
answered
so
your
much.
question. I hope I answered your question. Please say if I did not.
I understand. I just want to thank you for your time and effort and your need to be here and share your experience. So thank you so much.
Thank you for the opportunity.
Okay, thank you again, Tesha. And our final commenter is Joe Chobe. Joe?
Hello. Hi. I just want to take some time to thank ICER, Sickle, and everyone here that's able to participate. I'll give a brief background on myself. My name is Elena. I'm Joe Chobe. I was born in Togo in West Africa, and I migrated to the US when I was 16 years old. I was diagnosed at 8 months old with sickle cell SS. And since then, excuse me, today I'm 36 years old. I'm going to be 37 in October. And since then, I've had several crises, to say the least. So on average, prior to gene therapy, I was averaging about 14 hospitalization a year, right? And that's with severe VOCs. And each hospitalization could be from 5 to 14 days. And my longest hospital stay was 3 months. At the age of 19, I had my first hip replacement. 2 years later, I had a cord decompression on the second hip. And as you know, those procedures aren't cheap, right? So it takes a lot for someone that comes from a different country, for example, to go to school and try to live a productive life, but has to be in a hospital so many times a year. I was basically spending majority of my time in the hospital. And prior to, I'm going to skip a few things because we don't have much time, but prior to getting gene therapy, I took part of, to let you know which gene therapy I took part of was the Exocel from Vertex Pharmaceuticals. And I chose that because I studied both Vertex Pharmaceutical and Bluebird Bio. And I preferred the Vertex Pharmaceuticals because it was straight to the point and I didn't want to have any vector introduced to my body. So I took about 5 years to make a final decision really on where I wanted my life to go. Because like I was saying, prior to doing the transplant, I was hospitalized frequently. In order for me as an adult to maintain a quality of life, I had to get exchange transfusions every other week. And each exchange transfusion I had to receive from 6 to 8 bags, 8 units of blood. And those aren't cheap either. So imagine doing that every other week in order to just have a better quality of life, in order to work a part-time job and also own my own business. So it is important to have these therapies as they are evolving every day to the public, and for them to have a choice on what avenue they want to take when it comes to whether it's managing their everyday day-to-day life or wanting a better quality of life in general. So real quick, I wanted to go through some numbers. This is based on myself, right? Someone that has 14 hospitalizations a year, and each of them are 10 days each. That's the average. That's about 140 hospitalization days annually. And under the age of 25, living in the United States with a hip replacement at the age of 19, we probably get up to between $700,000 to $1.4 million within 25 years, right? And I'm 36 now. So if you imagine with all the organ damage, maintenance, the weekly infusion or the bi-weekly exchange transfusions, you're way above the 2 million mark or 2.8 million mark, right? And that's not even including if you have to seek therapy for mental health or anything like that. So is it worth it? If you ask me, yes, it's definitely worth it. It was worth it, especially for people like us that have severe sickle cell disease and don't have the opportunity like everybody else out there.
Joe, this has been very helpful for us, especially, as you said, sharing some of your numbers. I might have cut you off, and I'm sorry if I did, but
That's okay.
I hope you'll be here for the afternoon portion as well. And I really want to thank you for your comments and see whether anyone else has any comments or questions before we close for the morning. Jimmy.
Hey, Joe.
Hey, Jimmy, how are you?
Good, good. How are you, my friend?
Pretty good.
I think you did a fantastic job really giving us a look into what your life was like post-transplant. And I don't know if you can spend a few seconds telling us what your life is like now, at post-transplant rather, just to really highlight how fantastic this trial has been for you.
So I don't even know how to put it in words because I'm just grateful. It feels as if I have a second chance at life, right? Although I was pretty productive for the type of severity that I had when it comes to sickle cell crisis, I feel as if that now I can do way more. I run a business and manage 2 others and have a full-time job. I'm able to go to the gym, work out, take my wife out when I feel like it or when she wants to. But before, I couldn't do none of that. I could barely keep a job, So all I can say is that I'm very grateful that I was able to, undergo this gene therapy trial.
Thank you so much, Jimmy, for the question, and Joe. I'm going to turn it over to Steve.
Great, thank you, Rena, and thank you again to all the public commenters. A very rich topic, a lot to talk about already. We are going to have a curtailed lunch, but we are going to resume at the usual hour of 5 minutes past. So we'll all come back. It's about 35 minutes from now, and that's when we'll have further deliberation on the clinical evidence and cost effectiveness information and all these other perspectives leading up to some votes. And then we'll have the policy roundtable after that.